Good morning, everyone. Very warm welcome to all the participants joining us today for this engaging and much anticipated webinar, which is organized by National Medical Teachers Association. I am Dr. Rohan, and I'll be moderating today's session. We are gathered here to explore a cornerstone of modern medical progress. The topic is Basics in Foundation of Biomedical Research. Whether it is clinical trial or public health service or laboratory innovations, biomedical research is the backbone of evidence-based medicine. To guide us through this important topic, we are honored today to have with us Dr. Prakash Patel, sir. Dr. Patel is currently an assistant professor in the Department of Community Medicine at SMIMER. With over 13 years of teaching experience, he holds an MD in Preventive and Social Medicine and a PhD in Community Medicine, and has worked extensively across state health systems and national public health programs. He has been involved in major multicentric studies funded by WHO, ICMR, and Ministry of Health, tackling subjects like COVID-19 vaccine effectiveness, tuberculosis surveillance, and maternal and child health. His interest in health system costing and public health data has contributed meaningfully to national level policy discussions. Dr. Patel is also a prolific academician with dozens of publications in peer-reviewed journals and has guided multiple research initiatives from protocol design to statistical interpretations. Given today's topic, his insights into study design and data analysis and translational research will be invaluable, especially for young researchers. Now, before we begin, I would like to point out a few things. Please feel free to post your questions in the chat box. We will take them up during the question and answers session at the end. This webinar is being recorded and the video will be made available on our official YouTube channel. With that, I invite Dr. Prakash, sir, to begin his presentation.
Thank you, Rohan, for the invitation. Am I audible?
Yes, sir.
And are you able to see my screen?
Yes, sir.
Okay, so first slide is visible, foundation of biomedical.
Yes. Yes.
Okay. So thank you, Rohan, for the invitation, and also the Medical Teachers Association for the invitation. So, and thank you for my introduction. Okay. So today, what we aim today, we are going to basically it is an overview about biomedical research. We will briefly touch a few topics about the biomedical research. We might not able to go into detail about each topic, but we will briefly see what is biomedical research, what are the different type of biomedical research, what are the key steps in a research process, and a little bit about ethics, publications, and common challenges that usually a young researcher find while initiating their career in biomedical research. Okay, so let's start with what is biomedical research. If you see, by definition, it is a systematic investigation to understand the biological process and diseases. Okay, it is basically a biomedical research bridges gap between like clinician do a day-to-day clinical practice, and what are the basis for this clinical practice. Let's say I give a treatment, or I diagnose a patient with tuberculosis, or if it is any disease, I use certain tool to diagnose or tool to manage the patients. Okay, so what are the basis for this practice or for this method? That lies in the biomedical research. Whatever practice we do today, or whatever clinician practice in their day-to-day life, that is strongly based by the biomedical research. That's why we call this era as a evidence-based medicine. So the whatever medicine we practice, that is strongly backed by the biomedical research. So this include lab studies, clinical trial, epidemiological studies, and many things. Okay, so that is what biomedical research is. Now, why we do biomedical research? First, like as I have said, okay, today we practice a evidence-based medicine. So whatever practice we do, we need evidence to support that practice, whatever drug we prescribe, whatever surgical intervention we do, or what even for the diagnosis, we need certain evidence, and to generate this evidence, we need biomedical research. If we have a good evidence, we will have a good clinical outcome. If we support whatever practice we do, if it is supported by good biomedical evidence, biomedical research evidence, then the outcome will be better. The research also drives innovation in health and treatment. Like today in this era, daily we have a certain new knowledge or new evidence. So to update or to innovate in healthcare and treatment, we need biomedical research. So it's also innovation is also there when we do a biomedical research. It improve understanding of our disease mechanism, and ultimately it will help to improve the global health scenario. Whatever right now, whatever health situation we are facing, this research ultimately help to improve this scenario. If we go ahead, then let's say what are the different type of biomedical research. Like people divide this type of biomedical research in a different way, but if you want to just understand the basic things, which we can divide biomedical research into three categories. One is basic research, where we do a basic lab-based or even research at the cellular level or DNA level. That is basic research. Applied research means it is about the clinical practice or whatever problem right now we are facing in medicine, and to how to solve this problem. That is applied research. And then the third is translational research. That is bridging gap between basic research and applied research. How we can apply our basic research knowledge to improve the or how to apply it in a clinical practice. That is a translational research. It basically integrates between basic research and applied research. If you go a little bit in detail, so basic research is basically it's seeking the fundamental knowledge and understanding. So it more about theories and proving the theories or generating the theories. Okay, it is, or you can say it is a theory driven. Okay, and whatever impact you get, it is a long-term impact. Whatever, like let's say we are doing a research on studying a structure of DNA or molecular pathway of certain things or certain disease, then to apply this in a real practice, that will take a long time. And after that, like whatever we aim to improve the health, that will take a long time. So basic research has a good impact. Many times it is a huge impact, but it a long-term process. It take a long time for the impact. Applied research is it is about to solving the let's say practical problem right now we are facing. It is aimed to find the immediate solution or improvement in a real world issue. Let's say clinical is facing certain patient with certain sign and symptom. How to solve them, how to improve their or how to resolve their symptom, how to improve the quality of life. That is their aim. So it is a problem driven. What for problem we are facing and how to improve. So the impact is many time it is immediate. Okay, it is many time immediate impact or short, or you can get impact in a short term or in a medium term. Okay, let's say we are developing a new diagnostic case for a disease, for an existing disease. How to diagnose or develop a new test which easily diagnose, which quickly diagnose the disease. Now, translational research is bridging the gap between laboratory discoveries and clinical application. So whatever knowledge we gain during basic research. See, basic research is about generating a knowledge. Basic research, in basic research, we generate a knowledge. And to apply this knowledge into the clinical practice, that is a translational research. So it moves findings from bench to the bedside. So how we apply laboratory understand whatever we find in a lab, and how to apply in a clinical practice to improve the patient's life or to improve the clinical practice. Okay, it is application driven from lab to patient care. The impact, it is medium to long term impact. It will take time, but it is not a very far long. But uh, uh, it is uh, it is between basic research and applied research time frame. Like for an example, testing whether a new lab discovered molecule works in a human being or not, works to improve our understanding or not. Let's say you can say drug trial. Okay, so this is difference between basic, applied, and translational. Many a time, no such watertight compartments are there. Many research are overlapping in this the categories. If we move ahead, there are different domain of biomedical research. There are n number of domain in biomedical research. There are n number of areas where you can do a biomedical research. But if I briefly divide them, then you can, one is a laboratory based research, or let's say molecular research, biological research, genetics research, where we do research in a laboratory. The second type, second domain is clinical research, where we do research actually on the patients. Let's say clinical trial, drug trials. Okay, there is a clinical research where we involve the patient. Then next is epidemiological research, which is based on the population, which is done in the, let's say, in a community level. Okay, that is epidemiological research. Then the next is a health system research or health service research, where we try to assess the system or existing health system cost effectiveness, whether providing X services is a cost effective or not. Generally, this type of research are done by government, or it is initiated by government and ask research institute to do this type of research, whether it is a cost effective, whatever intervention government has decided, whether it is cost effective or not. Let's say there is a new vaccine. Okay, the research is done in laboratory to development of vaccine and effectiveness that we can do in a laboratory. Later on, it come to the clinical trial whether vaccine is effective or not. Once the vaccine proven effective and government start providing vaccine, or it is available in the market, later on we do certain epidemiological research whether it is in a real life situation whether the vaccine is effective or not. The vaccine might be involved or might be included in the government health program, and government want to do study whether vaccine is cost effective or not. Whatever disease we are prevented with this, it is cost effective in term of what resources are spent on vaccine development and the distribution of vaccine or not. So these are the different domain of biomedical research. It is not limited to laboratory. It is far and it is far beyond the clinical practice also. But the ultimate objective is to improve the quality of life or health of the community. Now, what are the steps of biomedical research? There are, like, most of people, those who have already done a small project or even large scale study, know this step. The first and the crucial step is to identify research questions. Okay, many people face the issue at this level. I don't know what to do, what to what should I take as a research topic. Okay. So this is the whole research started with this first question or the research question: what to do, what topic should I take. Okay. It is accompanying with the literature review. Literature review help you to find out. Literature review means searching what already people have done or what already people are doing. So to find out this, what are the gaps, what are study about the topic, and then decide the topic. That is the first step, and that many people find is the most difficult step as far as the research is concerned. Okay. Once you have decided the research question, next step is to frame an objective. Now, from framing an objective is also an important step. I would say it is the most important step. When you have decided the topic, the framing objective is the most important step for the whole research study. Everything else the next forthcoming steps all are depend on your objective. So the next step is to decide what objective or what we want to do. And then next is developing a protocol that is based on the objective, what we want to achieve. That is our objective, and how we are going to achieve, that is our protocol. So that includes study design, study tools, various methods, analysis plan, and that everything is in a study design. The next, once the study design or protocol is ready, then we will go for the approval of the institutional ethics committee or institutional review board. Once it is approved by the review board, we will start doing or conducting a study. So actual data collection start. After the data collection, it's time for analysis and report writing, where we frame our report based on the finding what we get. And then the final step is publication. So these are the key steps in research process, how the research is continued. I will briefly take each step one by one, and we will understand the importance of each step. Okay, the first and foremost thing is to decide a topic. Okay, and to decide a topic, it is very important to do a literature review. What is literature review? It is a systematic evaluation and interpretation of existing research relevant to a specific topic. Okay. So whatever knowledge is available on a particular topic, these systematically evaluate this existing knowledge. Okay. We systematically study what the research is going on, what are the recent articles published on this topic. So how it is helpful? It is helpful. First, it is helpful to decide whether I should take this topic or not. I have decided to find out some correlation, let's say correlation between hypertension and stress. Okay, but when I do a literature search, I could see that there are n number of publications available on the same topic from the even in my area, like I'm living in India and Gujarat, there are a lot of research from Gujarat also. Okay, so there is no meaning of proving the same thing which is already very well proven. So we have to move ahead, that there is no knowledge gap. The knowledge is already there. So no meaning of reinventing the wheel, doing again and again, there is no meaning. So we'll go ahead. Either you have to do something different. You may find that if you find that there are certain gaps in this research, then only you can go. So this will done how we will know this. This is based on the literature. I have seen many people don't do literature search much. They just take a topic and start doing research. And when they start writing a paper, at that time they start doing literature search and to write down the introduction or discussion section. But that is too late. You need to give some time, or say enough time for literature review, if you want to find out, if you want to take a good topic which ultimately lead to a good publication. Okay. Probably when you go for the publication, the editors will see this. If it is a common topic, lot of research available, then they will not even take the research. They don't go into research methodology. Just by looking at the topic, they will not take the topic. So I suggest to do a thorough review literature when you start developing your research topic. Why it is important? I already said it is able to identify the gap. It helps us to refine our research question. If something is already done, then we will little bit move ahead and do something else which is not done. When you do a literature search, let's say I am searching I am reading a research paper. You might have seen at the end of research paper, the author itself suggests certain future scope of research that I have already done, what is need to be done in the future. Usually many papers mention this. So that help you to define, refine your research question and prevent the duplication, which is already done. Okay. Literature searches also help you to develop design. It helps you to find out various methodology which you can implement or which people has already done. So it helps you to develop your design or it improve your design, whatever you are thinking. Okay. So literature review is very important. A strong literature review is a backbone of a research project. Okay. So for a successful research result, we need to do a good literature search. So I strongly recommend give enough time for literature search. Read what people have already done. That will help you to find out a better research question. The next is developing objectives. Okay. Say, as I have said, this is the most important part. Once you have decided the topic, objectives are most important part. It is a clear, concise statement that define what the researcher intended to achieve through the study. See, I say objective is everything. Objective is everything. The plan of the study or the research design, analysis, everything depend on the objective. What objective you are, how objective you are framing. Okay. It provide direction. So what objective you take, your research design will be based on that. Let's say I want to find out prevalence. So what type of study I should take? I should take a cross-sectional study, which help me to find out. I want to find out the incidence rate of something, then I need to take cohort. I need to take cohort study because you cannot find out incidence rate in a cross-sectional study. You need to go for a cohort study. Later on, we will see different type of study design. But what I mean to say, if you want to let's say find out the effectiveness of certain intervention, then you need to go, you have to design your study like a trial. Okay. Let's say drug trial. If you want to find out the effectiveness of a drug or different combination or different dose of the drug, then you should go for the design like a clinical trial. If you want to find out a correlation between two factors, then you should take any analytical study design, maybe case control study or cohort study. If you want to find out a risk factor of something, a risk factor of a disease, then you need to go for analytical design. It may be cohort study, it may be case control study, any type of analytical design. So what objective you take, the research design it is based on that. Okay, let's say I want to find out a risk factor of hypertension, then I have to take a research design where I take hypertensive people as well as non-hypertensive people and study what are the whatever risk factor I am taking. Let's say I am taking a smoking. So whether smoking rate is there what is the smoking rate in hypertensive people and non-hypertensive. So what is my objective? The whole design will be based on the objective. The objective clarify the boundaries. Okay, so clearly I have a scope. Like I am doing a study on a hypertensive patients, then whatever result I will get, that will be limited to hypertensive patients only. So it clearly define the boundaries. I am doing a study in an urban population, whatever result I will get, that is for urban population. I am doing a study, let's say, on infertile couple, then whatever result I will get, that is limited to that only. I am doing a study on children, that my result will be limited to children only. So it clearly defined the scope of the study. So next is facilitate planning, help in selecting appropriate methodology, tools, and sample size. Okay. So this steps like what tool I have to take to assess something. Let's say I want to assess a depression. So what tool should I take? That you have to clearly mention in the objective. There are n number of ways to assessing depression. So what tool I use, that you have to clearly mention in that I will use dash score or any other score. So that you clearly mention in the objective. So what tool you have to take, that is your objective will tell you. If you have properly designed your objective, then this will very helpful in selecting the tool. Sample size also depend on the type of, like it is sample size basically depend on type of study. And type of study basically depend on what objective you want to take. So it is also helpful in designing the whole methodology, including the tools and sample size and everything. It also helpful to measure the specific outcome. It that is at what we how we define it in a methodology. It also helps to enhance communication, like clearly convey the purpose of study. It clearly, the good objective clearly convey the purpose of study. Let's say when I start reading any paper, any manuscript, what I first will look at the objectives. Suppose you are presenting your study in an ethics committee. What the ethics committee will ask you, tell me your objective. Okay, so objective convey in a one sentence, one or two sentence, objective will convey what exactly you are doing. When you present protocol to ethics committee or even any funding agency, they will assess whole your protocol based on your objective. What objective you have selected, and based on that, they will assess whether the study design is in a line with your objective or not. Okay, when somebody review your paper, let's say I have a publication, I have submitted my article, at the end of my research, I have submitted an article, and it goes to reviewer. Now, how reviewer will look at your paper? They will look in context of your objective. What are your objective? What you have decided to do first, whether methodology is in line with your objective or not, and then see whether the findings are in line with your objective or not. Whether, like, let's say simple example, you have decided to find out prevalence, whether your result shows prevalence of whatever you have decided, whether you have decided to study the prevalence of hypertension, whether your result shows prevalence of hypertension or not. You decided to study correlation between these two factors, whether your result shows correlation between these two factors or not. So it clearly convey what you are going to do or what you have going to done. Okay, even your conclusion of your study. You see, many people ask how to write conclusion. So conclusion is nothing but it is the answer of your research question. What research question you have stated earlier, answer of that research question is your conclusion. So what you conclude at the end of study, I want to find out, I want answer of this question. At the end of study, in what I write in conclusion, what is the answer of that question? That I write in conclusion. So what I mean to say, everything depend on objective. That's why I say objectives, framing and objective is the most important part of any study design. And it also helps to support the ethical approval or ethics committee approval. Many times people use this acronym, SMART. The objective should be SMART. It should be specific, measurable, achievable, relevant, and it should be a time bound. So whenever we frame objective, we need to see this, whether our objective meet this criteria or not. Then next is a study design. What we have decided in our objective, and then based on that we will develop a study design. There are a lot of different type of study design. I am not going to go into detail about each and every type. But what I mean to say, it is a blueprint of your research validity and reliability. The research design will tell you whether your study is valid or reliable or not. So, like external person will assess your research based on your research design. If it is a thorough, if you it is in detail and thorough, then it may be a good research, good, valid, and reliable research. Okay. See, the findings matters, but whatever findings come, whether it is positive or negative, that doesn't matter. What matter is how thoroughly you have done the study, and that depend on your research design. So it is the finding, whether it is positive or not, that is not important, but your research design, whether it is good or not, that is important. And if you have a thorough research design, if you have a good research design, whatever finding comes, that will be acceptable by everyone. Okay. You cannot presume what finding I will get. Okay. But what is in your result are not in your control, whether it come positive or not. But research design is in your control. If you develop a good research design, whatever finding you come, if you have a like, let's say I have a negative finding. Most of people have a positive finding for certain things, and I have a negative finding. So many people say that it is not good. No, it is not. It is nothing like that. Negative findings are also acceptable, but you have to back your negative finding with a strong study design. So it is the most important thing, study design. It is a blueprint of research validity. Okay, you have to define, you have to design research in such a way that minimize the biases. Uh, Maybe future in a future session we will discuss about bias and everything, but you have to design a study in a such a way that it reduces the bias or minimizes the bias. Okay. Uh, if we briefly touch the study design, basically we divide the study design into two categories. One is observational and one is a experimental study design. Okay. So whatever like observational means we doesn't interfere with the natural path of disease. We just observe what is going on. That is observational study. And experimental study means we try to change the path, the natural course of the disease. Okay. That is experimental study. Simply RCTs, randomized control trial. We give drug and try to modify the path whether the disease symptom improve or not. That is experimental study. And observational studies, these are non-interventional study where we just observe what is happening. Let's say the observational study are divided into two groups. One is descriptive study and one is analytical study. Descriptive studies are usually cross-sectional. It may be a longitudinal, but it is usually a cross-sectional study where we describe the disease. Let's say I want to see, give me a simple example. I want to find out the prevalence of hypertension in medical faculties. Okay. So that this is a cross-sectional study. Once I will go to all the faculty and measure their blood pressure, whatever method I have decided to assess the blood pressure, that by using that method or by whatever definition, I will assess their blood pressure and decide whether they are hypertensive or not. Okay. This is a cross-sectional study. At the end of study, I will able to know what is the prevalence. It's 10% people are suffering from hypertension, 20% people are suffering from hypertension, like that. This is a descriptive study. Okay. Here I describe the disease or whatever my topic, I describe in term of time, place, and person. I will describe the condition like in how many, whether it is higher in male or female. Let's say I have done a study on prevalence of hypertension in medical faculty. I will take male and see whether the prevalence is there in what is the prevalence is in male and what is the prevalence in female. So this is a descriptive study. That study, descriptive study, help us to find out the hypothesis. At the end of study, by comparing other studies or the comparing prevalence of hypertension with in the other group, I can formulate a hypothesis whether the hypertension, let's say prevalence of hypertension is higher in medical faculties compared to other professionals. That would be my hypothesis. At the end of study, I can generate that type of hypothesis. How now I want to test this hypothesis, whether it is true or not. That I can do in analytical studies. So in analytical study, we will always have a group. We will always have a group at the beginning, and we compare in this. Let's say I want to compare hypertension, see whether prevalence of hypertension is higher in medical faculties compared to engineering faculty. Then I will take a medical faculty in one group and engineering faculty in another group, and then measure their blood pressure and compare them. So that is an analytical study. Okay, that what I am saying is very basic study, just for an example. Okay, if we there is basically two type of analyticals. There are many types, but basically two type of analytical studies are commonly used. One is case control study and cohort study. Now, what is the difference between case control and cohort study? Case control study start with the disease present and disease absent group, or disease or whatever health condition I am taking. Let's say patient with lung cancer and patient without lung cancer. That study start with that, and then we go into the past record or past history of the patient and see what are the risk factor present in patient with lung cancer group, and what are the risk factor or what are the level of risk factor present in patient without lung cancer, and then compare them. That is a case control study. At the end of that study, we get odds ratio. Okay. Cohort study start with the risk factor. How many people are smoker? There are group of people who are smoker, and this is a group of people who are non-smoker. And then we follow up them for a certain period of time and see whether they develop lung cancer or not. That is a cohort study. Okay, so cohort study is from risk factor to outcome, while case control study is outcome to risk factor or disease to risk factor. Okay, but in short, we doesn't interfere the natural course. Whatever is happening, we just observe them. We divide them into a group and then observe. We take them as a single group and then observe. We observe in a retrospective manner, or we observe in a prospective manner. That is a different way of observing, but that is ultimately we observe the things. That is observational study. And when we change the course of natural course of disease or certain natural pathway and try to modify the things, that is experimental study. Okay, if you take science, if you take this study in a term of value, which study is higher value, then experimental studies are, you can say it is like a gold standard. There's highest value. If something is evident from RCT, then it has a highest value. It is followed by analytical studies and then by the observational study. So observational study has a least value if you put them in a evidence pyramid. Okay. That is observational study, and that is after that analytical study, and after that experimental studies. Okay, you might have heard about meta-analysis and the systematic review. That is above the experimental studies. Okay, but this evidence, the value of evidence depend upon the how scientifically the study design is well. If you have proven something by experimental study, but the study design has a lot of loophole, then it has a very less value. Okay, so don't just take a study design as don't just consider study design, but also the type of study. Go into detail of research design and see whether the design is scientifically valid to prove the whatever conclusion they have drawn. Okay, poorly conducted analytical study, good analytical study is much better than poorly conducted experimental study. Okay, but most of the time the studies start with the observational study. People observe something. Even before observational study, you have case reports. Many clinical research start with a single or a case report and case series, and then it converted to the observational study, and then people try to do analytical study, and then finally experimental studies come. Okay. So experimental study has a very good value, but it is not like that you start with the experimental study. Okay. So these are the different type. Each study has its strength and limitation. Let's say cross-sectional study, it can't interfere causality. Causality means what comes first. Like today, let's say I want to see hypertension and stress. Whether I have done a study and cross-sectional study, and I have assessed hypertension and also measures the stress level in that population where I have studied the hypertension. Okay, so in a one person I have assessed blood pressure level as well as the stress level. Okay. Based on these things, I cannot tell. Suppose I find that the stress is strongly associated with the hypertension, but based on this study, I cannot tell that the stress comes first or the stress lead to hypertension or hypertension lead to stress. That causality I cannot interfere based on the cross-sectional study. So if your objective is to prove causality, then you cannot take cross-sectional study. I have to find out risk factor is causality. Risk factor means suppose smoking is I want to prove that smoking is a risk factor for lung cancer. then smoking should start first and then lung cancer should come. Okay, so in that case, I cannot take a cross-sectional study. Okay, so cross-sectional studies means we are measuring both the things at the same time. So cross-sectional study doesn't help. It can't interfere causality. So if you want to prove causality, you need to go for analytical study, case control or cohort study. That help you to prove causality. What comes first? Okay. Case control study, as I have said, you start with the disease present and disease absent, or whatever outcome you take, that is present and absent, and then you go into the past of the patient or record of the patient and study whether the risk factor are there or not. So we already have a disease people, so it is good for rare disease. You take the rare disease people and then take the control group and see whether risk factor are there or not. Okay. But recall bias is the biggest issue with the case control study, because we are taking data from the past. So when we go into the past, recall bias is a major issue. It is like commonly what happens, those who are suffering from disease, they better know their past, or they better know what is happening in the past. Suppose like I am suffering from a diarrhea, so I will better recall what I have eaten in last few days. Okay, if I am not suffering, I might not recall, or I might not try to recall it. So recall bias is the biggest issue. When you go for cohort study, it is it has much better value than case control study in terms of cause to effect. When you want to prove that this is a cause of certain outcome. Okay, but it is lot of time consuming. It take lot of time because it is a usually it is a prospective study. We go in a prospective manner and see whether the person has developed disease or not. Let's say smoking and lung cancer. I have to follow many years. For many years, I have to follow a smoker for many years to see whether they'll develop lung cancer or not. Because development of lung cancer takes a long time period. Okay, so usually cohort study are very expensive. It is difficult to do a long follow-up of the same patients. Okay, so it has every study has a pros and cons. There are different ways of doing, like you might have heard about retrospective cohort study, prospective retrospective cohort study, that we will discuss maybe in some other session. The next is RCT. As I have said, is a gold standard, but it is costly, and there are a lot of ethical issues associated with RCT. And now, as as our knowledge regarding the human right increases, these issues come. Many a time it is a lot of problem as far as the RCT is concerned. Okay, but it is still a gold standard. Meta-analysis, systematic review, all these are primary studies where you actually collect the data. Meta-analysis and systematic reviews are secondary studies. They are based on somebody else's study. Okay, so those they have a good value, but that is not a primary study. Okay, and then next comes the when you have decided a study design, your protocol is ready, then comes the approval of ethics committee. So role of the ethics in biomedical is very important. Like it is our moral responsibility to protect the rights of the patient, not only rights and dignity of the patient. So you have to design your research in such a way that it is ethically correct. Okay. Scientific correctness is more is primary. Like first you have to design a study which is scientifically sound, and then this study should also be ethically correct. So you have to frame let's talk about the consent. You have to develop your consent from patient information sheet and consent form in such a way that you communicate what you are going to do with the patient, and let the patient decide whether to take part in the study or not. So you communicate properly so that you keep your study transparent. You become honest while communicating to the patient. Tell the patient what you are going to do, what is all about this study, what we are going to get at the end of study, Whether you get something or not, that also tell the patients. Okay, don't give much incentive. So at some time incentive are important, but it, it the incentive should not be a large enough that patient participate in the study just only for the incentive. Okay, so the participation should be purely voluntary, and to take voluntary decision, tell the patients what you are going to do. Okay, that also help you to build the trust between this researchers and the participants. And if patient has a trust on you, you will get a better result. Okay. And if we keep the like moral issues and ethical responsibility, if you keep aside, this nowadays it is also a requirement. Every human research must have to take approval from the institutional ethics committee, and that ethics committee should be registered to the government, unlike let's say ICMR or DHR. Okay, so it is a requirement as per the guideline also, and it is also our moral, or even as a scientist, it is our responsibility to maintain highest ethical level in our research. Okay. Uh, what key principle we need to keep in mind? That is autonomy, maximize benefit, do not harm, fair treatment. All this you might have heard at some one or other point of time. But what I mean to say, whatever participant take part in the study, their participation must be on a voluntary basis. You need to take consent, or you need to take transparent consent. So, like if you are taking a children, then you need to take consent of not only the consent of the parents, but you also need to take assent from the children. When you take a illiterate participant, then you have to ask somebody to be a witness of the consent form. There is, like many people mention in their study about the oral consent or verbal consent. There is nothing like a verbal consent. If you are doing a verbal consent, then you need to record that consent. Just mentioning that I have taken verbal consent, that is not enough. Consent is always written. If you are taking a verbal consent, then you need to take permission from the ethics committee and need to record the consent. You need to record that process, either audio-visual recording or just audio recording. And that is evidence. You need to keep some evidence that patient has given, and that is not only for the participant, that is also for the researchers also. Might be happen that in a future date, the participant might claim that the researcher has involved me in the study without my consent. I have not given consent. In that case, you have evidence that we have taken this consent. So consent is for both the boys, participant or patient, as well as the researchers. So you need to keep this in mind. Role of the ethics committee in protocol review. Ethics committee also has a very well defined role. Okay, ethics committee will see whether whatever consent form you have prepared, whatever the process you have stated for consent, whether it is correct or not. Like you are a treating physician and you are yourself taking consent of the patient. That is not correct thing. Like a treating physician, how a treating physician can take consent? Patient is in obligation of a treating physician. This consent, most probably it is not a voluntary consent. Okay, suppose one teacher who is teaching a student and taking consent to take part in the study. This consent cannot be a voluntary, because the student academic performance depend on the teacher, and that teacher is taking consent. Then how can it is a fair consent or a voluntary consent? So there are certain process which you can adopt to avoid certain that, like most of the time clinicians are like, suppose clinician who is treating physician and doing a research on a patient. So he can assign taking consent task to somebody else, and that third person come or let's say co-investigator or somebody else into the study which come and take the consent and then send the patient to the physician after taking consent. So at that point of time, patient, person who is taking the consent is not a treating physician. So you might expect some voluntary participation, or patient may have an option to deny. But if a treating physician asks, then how patient can deny? So this process, how you are going to take consent, is very important. So whenever you develop your study design, you mention this process, how you are going to take consent. It should be transparent and fair, and ethically, it should be the process should be ethically sound. The consent process. Okay. So this is all about the ethical principles. Then next come the data analysis. Once you have conducted you have uh, presented your protocol in the ethics committee, and then you have conducted the study, whatever tool and protocol you have decided, you have done the study, and next come the data analysis. Okay, so I am not going to tell you how we are going to do a data analysis. That may be a later on some other day. But what is important to tell you, that whenever you do analysis, be non-judgmental. Don't be judgmental while doing analysis. Like, let's see, don't presume the findings. Many a time people think that they are already assumed that I want to this type of findings. I'm going to get this type of finding. That will hamper the analysis. Okay. What you have to do, you have to be objective. Prevent personal bias from affecting the interpretation. Whatever interpretation, see, study design is very important. Everybody will see your result in context of your study design. So if you have a good study design, don't bother about whether the findings are positive or not negative. Okay. If we are going to get positive finding every time, then there is no meaning of doing study. Okay. The path-breaking studies are always different than what usually people find. Okay, so don't afraid about this negative finding, but be sure about your methodology. If your methodology is good, then the finding, whatever finding, that are good finding. Okay, value all outcome, negative or non-significant result are all equally important. Okay, suppose I find that smoking is not associated well with lung cancer. There is a very important finding if my methodology is good. So non-significant finding or negative finding are also important. Afford the research integrity, honest reporting the strength, the credibility of science. Okay, so whenever we present our finding, we always, along with that, we always put strength and limitation of our finding. So whatever analysis or whatever conclusion we draw from the study, before that, accept what are the strength and what are the limitations of our finding. Okay, so that help you to present your result. Suppose there is a something which is limited to you, like let's say a simple example, when if you are doing a study in medical college, some resident is doing a study in a medical college and taken very less or very few samples, then accept that in your before drawing your conclusion, accept this, that the limited number of sample is one of the limitation of my study. If we go for a large sample, then you have a better risk, but accept that this is the limitation. So whoever reading your study will not comment that the sample size is less. They will see, okay, author has already accepted that the sample size is less. So here, then the reviewer will see beyond that number of sample. Okay. So whatever strength and limitation, honestly accept that while presenting your finding. Let the data speak to itself. Analyze finding with curiosity, not expectation. Don't expect that my result will be positive or negative. Just, I want to see what is going to come. That curiosity, develop that curiosity and try to find the answer of that curiosity, not your expectation, whether it should be a positive. Many residents come to me while doing analysis that after the analysis, then it should come statistically significant. If we have already decided that the result should be statistically significant, then there is no meaning of doing the study. Okay. So whatever your result, you see it with curiosity, not the expectation. Okay, and then the final step, publication. Okay, see, it is said that unpublished research is unfinished research. If you are not going to publish the research, then there is no meaning of doing the research. Okay, so publications is the final step, and without that, the research is not, like, it is unfinished. Okay, why we do publication? Because ultimately, we have the research. Ultimate aim of the research is to fill up the gap, the knowledge gap which we find before the start of the study. We need to fill up the gap. How you fill up the day? You tell everybody that I have done study and I find this. Okay, so dissemination of knowledge is the purpose of publications. It promote the scientific progress. You have done something in this field, then next person will go something a little step, one step ahead, and advance the scientific progress. It also ensures the accountability and transparency. It contributes to the evidence-based practice. How we get evidence, how we know that the evidence are there, that is from the publications. I want to find out evidence about something, so I will search the publications. So it is important when we are practicing evidence-based medicine. It also enhance your academic reputation, like how, like whether you are a good researcher or not. How I come to know, that is based on your publications. So it enhance your academic reputation also, and it also fulfill the ethical responsibility. Okay, when we do a such study, it is my ethical responsibility to publish this finding, to send this finding to the public. Okay, so I have to, and in a way, to the participant also. In a way, this is for the participants also. So I am telling the participant as well as the public and scientific community that what result I get. So publication is very important. And see, publication, like there is a whole separate session about publication, but whenever it is important to do a publication, not only publication, but publication in a good journal. That is, nowadays predatory journals are very common. So we are talking about publication, but not only the publication, but publication is in a good research journal. Okay. So this is whole about what, like, from the where we begin our research, and the publication is the end point of that research, and somebody or you yourself can go ahead and do further research. Now, I have listed out some common challenges. There are a lot of challenges in biomedical research, and specifically when you start your career into that. But there are common biomedical challenges in this field. The first is formulating a focused question. That is the most important challenge many people face. Okay, how to formulate a good research question. What I suggest, read as many as paper as possible. Try to read, and not only read, but read from good journal. Take high impact journal. See, PubMed index, Scopus index, Web of Index, that doesn't matter. You take a journal, try to find out the impact factor of the journal, and then read that. Reading the article will help you, helps a lot to formulating a correct or focused question. Choosing an appropriate design. This is also many times a challenge, but specifically when you don't understand about your research design. So I will suggest that before selecting a design, go through various research design. Go and that literature service help you. Go through a lot of paper with different type of study design. That help you to choose the correct research design for your whatever objective you are deciding. Lack of research training, that is also a challenge for where to get the research training. See, biomedical research, basic course of biomedical research is one area where you can begin there are very wonderful books available where you can read that books to improve your knowledge regarding the research. So this you need to take a formal training for research, or you need to read a lot about the research to get better idea about the research. So you have to involve in the process, and gradually you will get gain a knowledge on biomedical research. Time and mentorship constraint, that is also a constraint. Time constraint, uh, mentorship constraint. See, finding a good mentor is like, it is very difficult task, very difficult. But go if you have a, like somebody in your department or in your other institute who is, who has done a lot of research or done a good research design. The number of research is not important, but what type of research they have done, what is the quality of the research project that they have done, that will help. Like if you find somebody who has this type of experience, that then it will save you a lot of time uh, in this field. Limited funding or publishing support. Limited funding, uh, actually limited funding is, I accept, is a challenge, okay, but it is not an issue. There are a lot of agency who are ready to fund you, but but you need to go, or a lot of agency, we are ready to support you also for publishing. Nowadays most of the journal are paid journals, so you need a certain financial support. But for that to have a funding or publishing support, you need to have a good study. You need to have a good study, or let's say a good research question and research design when you apply for the funding. Okay. There are a lot of funding agency, like including IMR, DHR. Then there are many other international agencies who are ready to fund, but the condition is that you have a good research study design. So invest your time in studying the research design, literature review. Then you have a good design or good study. And then the last challenge is publication. Many people find challenges while publications. Okay, but what I say, if you have a good study, then the journal are very much interested in publishing your article. Okay, they have to publish certain things, but they are interested in a good research, good study design. So whenever you go for the study design, but don't select a journal who is quickly published, but go with the impact factor. Which journal has a higher impact factor? You start with that journal. Many a time, the reviewer or the critics of the article will help you to improve your research design or improve your article. So let them criticize your research or your article. Let's send it to the good journal. Let them criticize. It will definitely improve your article. Uh, and then improve your article and go for publication again. So it's a time-taking process, but publishing in a good journal is very important. It is it validate your study, it validate your research, and also help you academically to like improve your careers. Okay. So these are the common challenges that people face. There are certain misconceptions, specifically for initial researchers. Led research is only for MD and PhD. It is not like that. Everybody can do research. Even nowadays, undergraduate students also involve themselves in a research. You need big funding. That is not many cross-sectional studies or many observational studies we can do even without funding also. Okay. Or it can be done by a small amount of fund also. Okay. For publications, there are journal which publishes article free of cost. Okay. So it is not like that everywhere funding is required, but your scientific content should be at par with their expectation. Then only they will take. Case report doesn't count. Like case report are also important. Case report and case series are also. Many time research studies start with the case report. Negative results are failure. That is a misconception. I already said. Result, whether it is positive or negative, that is not important. What is important is your methodology. If your methodology and objective is good enough, then result also counts. Ethical approval isn't needed for survey. See, every research which involves human being required ethical approval. That require ethics approval or institutional ethics committee. Okay, so even every every research, whatever research you are going to do, you need to take approval from the ethics committee. Okay, whether it required consent or not, that is a secondary thing. Uh, you ask the committee uh, and take a waiver for consent. Let's say you are doing a study purely on lab base, you have a sample, you are doing study on that. You ask the ethics committee that we want a waiver in this case because we are not directly dealing with the patient. In that case, ethics committee will see your protocol and may grant you a waiver. Okay, but ethics approval, even just for a simple interview type of study, you need ethics approval. Any research which involves human being needs approval from human ethics committee. Okay, so these are some sort of misconception. Okay, so this is the end of this is the last slide. What I want to say, I have told you. So basically, to summarize this, we are in an era of evidence-based medicine, and for that we need to do the biomedical research is very important. All the evidence are driven by the biomedical research, and to do biomedical research, objectives and research design are very important. So invest your time in studying this literature review and the study design. Okay. And then develop and follow the structural process. Whatever process we discuss, that is just a brief, but whatever structural process is there, you need to follow each and every aspect of that, follow or uphold the ethical principle at every step. That is a most important part when we most important parts related to ethics when we talk about a human research. So that you need to uphold this at every step, and you don't need to start at big, but start right. Find out the right question, find out the right design, and then start the study. It may be a simple observational study, it may be a simple cross-sectional study, but you need to start. Just start it and start right. So that is what I want to convey from my side. Now, if anybody has any question, then they can ask question. Now it's time for question and answer. Rohan?
Yes, sir. Sir, there was one question in the chat box, but I think you have addressed it already in the webinar in the last 2 slides. So I think it is addressed now.
Okay.
We can ask for any other questions from the candidates. The candidates can unmute their microphone and ask to sir, or either they can write in the chat box also. Yes, so I think there are no questions, sir.
Okay. So alternatively, you can share my email address if anybody wants to.
There has been one question, sir, from Dr.
Yes.
Patel. Which is the most difficult part of any research?
As far as like, if you want my opinion, finding a research question and developing objective is the most difficult part. If you have a good question, then everything you can take help from other people to develop a study design, study tool and everything. But like to find a research question, that is the most difficult, that you yourself have to do. Like doing a study on somebody else's research question doesn't help you a lot in developing your research. You find it yourself, and that is the most important part as far as like my opinion is concerned.
Yes, sir. So this brings us to the conclusion of our today's session. On behalf of NMTA and the entire organizing team, I would like to extend our heartfelt thanks to Dr. Prakash sir for delivering clear, practical and comprehensive topic. I hope today's session inspires our candidates to pursue their own research journeys with greater clarity and confidence. Thank you so much, sir.
Thank you very much for the invitation. Thank you.